well, this whole panel here, I'll tell you, is very passionate about their own pets and stuff like that. I want to start off with showing my pet. This is Dash. Okay? I love Dash. I don't want to see anything come bad to him and stuff like that. He's a trail dog, okay? I'm a runner. I want him to go running with me. Well, I'll go running 25 miles. He runs 50 miles, okay? Because he's running after every rabbit he sees and things like that. So, and then when we get all done and I'm tired, he still wants to go running more, cries that he wants to do more. He also likes going to work with me. I mean, we have fun there and stuff like that, doing experiments and stuff. But I spend a lot of time with him, and we've also bonded quite well. As I said, I love this dog. It's Dash. Now the problem is, they start off small, and they get big, and then big is okay, but then they get old, and dogs have a very short lifespan. This is Dash now. That's what he was just 4 years ago. He just turned 4 years old. And I'm always depressed thinking how short their lifespans are. This is a chart showing how long dogs live. And what I want to show you is Chihuahuas might be one of the longest living dogs, living 17 plus years. Now, everybody has stories of how they've had dogs live to be in their 20s and stuff like that, and that's because of really good care. But the average Chihuahua lives to be about 17 years. One of the nicest dogs in the world, Great Danes, only live to be 9 years at the most. Here it shows 7 years. Dash is right there about in the middle. His life expectancy is about 13 years. He was born 40 years after I was born. He's going to die of old age, statistically, about 10 years before I do, 10 to 20 years before I do. I mean, that's a crime. These animals are the greatest animals of all time. Okay, so today's panelists, we're all going to be talking about this, is me. I'm the CEO of Sierra Sciences. That's our website. Then Liz Parrish. She's the CEO of BioViva, and there's that website. Greg Fay will also be talking. He's the Chief Scientific Officer of Intervene Immune. He's also the Executive Director of 21st Century Medicine. And then we have Yuri Dagan, who's the CEO of Ethereum Genetics. And so all of us will be talking about our pets and the work we're doing to try to make them live longer and healthier. So there's others that aren't here that are also doing this. Alex German, he's working more on lifestyle. I recommend you Google him and find out information about him, about how to feed your dog, what kind of lifestyles to give him to make him live as long as possible. Then there's also, I don't know how to pronounce their last names, Daniel Promislow, Matt Cabrian, and George Church. They're doing work with rapamycin to look at seeing if rapamycin can extend dogs. They might be here next year. I hope everybody likes this panel and we can make this an annual event. My focus, of course, is on telomeres, and there's been papers showing that telomere length does affect lifespan of dogs. This is a graph just showing going from one end to the other of the shortest living dogs to the longest living dogs. I'll get rid of the pictures of the dogs there and show the data. It just shows that their telomeres, the longer living dogs have longer telomeres than the shorter living dogs, suggesting that telomeres actually play a role. Well, even a better paper came out looking at all different animals, and they looked at what causes aging in terms of telomeres in a lot of animals, and it turned out most animals on this planet do not age by telomere shortening. Some have no detectable aging at all. I could spend an hour talking about that, but what I want to show is that here's a list of the animals that have been shown to age by telomere shortening. That includes dogs, cats, horses, sheep, pig, deer, non-human primates, and humans. Those are the only animals so far that have been shown by age by telomere shortening. Most rodents, or all rodents, don't. It's something to do most of the experiments done on mice. They've had to engineer them. Well, we have developed gene therapies. I know I didn't go into exactly what gene therapy, and I'm looking forward to you listening to Liz talk about what that is. We've already developed gene therapies for dogs, cats, horses, and pigs. And the arrows are not quite right where they're supposed to be. But we have those sitting on the shelf ready to be tested in animals now, in pets. Oh, and primates, of course, which is humans. And we also have a lot of supplements. Nicola was speaking the other day, maybe it was yesterday, about something she called systems pharmacology. We've been doing that for 25 years. We call it medicinal chemistry and high throughput screening. And we've actually come up with supplements that actually do induce telomerase, but not as good as the gene therapy stuff does, but it at least would decrease the rate of aging in the animals. So these are all available, ready to go now. I want to show you this animal, because what's the problem with all those animals I just showed is that they're so big, and clinical studies, or preclinical studies in this case, are so expensive that doing a small animal is really beneficial, and that's why most people do their studies on mice. But mice don't really age by telomere shortening. It's very, very different. Well, I've learned that there's this mouse, or not mouse, it's a lemur, it's a primate called the Madame Bertha's mouse lemur. It's showing the smallest primate in the world, probably being held by the largest primate in the world. And it's only found in Madagascar. And so we have been in contact with the officials in Madagascar to see if we can start doing these studies on these different primates to see if we can make them live longer. One of the things that we have coming up, hopefully really soon, is something that we can sprinkle onto dog food, cat food, horse hay, whatever. We're calling it Telo Dash. It'll be one of our strongest supplements, and that's why we're calling it Telo Dash. All right, and thank you very much. Again, Sierra Sciences, Defy Time, Labella are my partners on this, and I would now like to introduce Liz Parrish.
Thank you. It's great to be here, and it's actually interesting to do this talk because we only recently planned to do this, and being the CEO of BioViva, we're committed to lengthening human lifespan and health span through gene therapy. And our exclusive partner to the company is Integrative Health Systems. So you might have seen the booth out there. They're offering consultations if you're interested in finding out if you're a candidate for gene therapy. But they not only do just gene therapy in humans anymore, we actually do gene therapy in pets. Because we know that they're your best friend, we know that they extend your lifespan, they reduce your blood pressure, and do all sorts of amazing things, and they're part of your family, whether you consider them fur babies or anything else. This is my daughter's newly adopted cat, and the photo's done by Steve Bartlett, which I have to thank him. He's here taking photos as well. And we want our pets to live a long time, and she wants her pet to live a long time. This is her first child, and this is my grandchild, my first grandchild, so I'm really excited about that. And so in order to meet that mission, we're specifically looking at gene therapy. I think that all of the talks here have talked about, our steps to making humans live longer. But we know in order to do it in a more permanent way, to self-evolve into a new future for mankind, we need to use genetics. And so this is my specific focus of my company and every company that we work with. And how gene therapy works is, later today I'll show you a video about it, but you put the gene of choice, the therapeutic gene, into a vector delivery system, and we inject that into the body. We might inject it directly into an organ or give it by IV and have it work systemically. And what happens is that vector attaches to the cell and it injects the therapeutic gene. And you might wonder if that actually works. Well, it does. Today, through the US FDA and the EU, there are 5 past gene therapies, and so these are one-time treatments for a lifetime, or at least they're considered that today. So in a decade or two, we'll find out if patients need another treatment, but you can imagine how this is going to revolutionize your life. Instead of having to remember to take pills, you literally will go in for a series of injections at different stages of your life to ensure that you don't get sick with various diseases. So in animal studies today, as far as the success rate in gene therapies, we see that VEGF 164 is actually working for lameness in horses. It's actually reversing lameness permanently in the joints of horses. Dogs, a lot of people don't know that the most common blood disorder in dogs is hemophilia A. And there is now a treatment for hemophilia A in dogs. And remember, these are fantastic models for humans. There are actually 2 treatments about to be passed, one for hemophilia B, and hemophilia A in humans is doing quite well in gene therapy as well. Also, dogs get something called an X-linked muscular dystrophy, and it's a really decent model for Duchenne's muscular dystrophy. And what they've done is they've actually upregulated the utrophin gene through gene therapy, and it is a curative effect. And also, so cats, well, what about our cats? AAV8 hex gene is actually cured Sandhoff's disease. And so that is showing you the power of the gene therapy. As far as cancer treatments, the IL-12 therapy is curing cancers and has been proven safe and effective in cats, dogs, and horses. Isn't that fantastic? So what are we doing at Integrative Health Systems? So you know, just like with humans, if you have a rare disorder, we are willing to make a gene therapy for you if the doctor agrees to do it, and do a treatment for any monogenic disease that there is a known gene therapy for. But we are known specifically for treating biological aging with genes that upregulate regeneration. So aging is cellular degeneration over time, and curing aging means that we need to upregulate regeneration over the degeneration. So we look at the telomerase-inducing gene. This is something that Bill has talked extensively about, and we know that it's the rate of shortening in the animals that are affected by telomere shortening that ends their life. So it's the rate of shortening that really defines lifespan. And so we definitely know that this is one of the keys to treating biological aging. We're also looking at muscle mass. So when you increase muscle mass, you actually create better insulin sensitivity, and you create a more robust body. And actually the gene that we're using today in humans and in animals, just in a recent paper just in September, it showed that the upregulation of this gene actually has a protective effect against cancer by stimulating the natural killer cells. So this is kind of a dual-purpose gene therapy, and we think that it will have a great impact on metabolic disorder. And so pets also go through a system of sarcopenia like humans do, muscle wasting over time. And for every 100 grams of muscle mass that your cat loses, they get a 20% increase of mortality. I just lost a 23-year-old cat, and he was pretty atrophied towards the end. And I was so sad because we started this company and got our vet on board right about the time that he couldn't travel anymore. He would have died on the trip. Clotho. Clotho is the goddess that spins the web of life, and this gene is absolutely amazing. Not only is this gene in studies showing that it protects against chronic kidney disease and cardiovascular disease, it actually is the #1 gene right now, gene candidate for association with intelligence. And as a matter of fact, when they took models of mice, both young, old, and a model for Alzheimer's, and injected them with the protein, they performed better on cognitive tests within a matter of hours. So smarter pets, healthier pets, stronger pets. Good stuff. This is my recent adoption. This cat's name is Lyra, and she's quite lovely. She hates the other cat that my daughter just got, just so you know. Not working out really well, but it's okay. Anyway, right now, the doctor that we use specifically for the gene therapies has a lot of background in stem cells and PRP, and these work really well. They're helping lameness in horses. They're getting them back on the racetrack. They're helping cats with asthma, kidney disease, and intestinal disorders, and dogs with various disorders as well. And I'm going to show you some videos of some of the work that our vet has been doing that works for Integrative Health Systems pets. So this dog on the left has hip dysplasia, and it went through a series of PRP and stem cell treatments, and we can see here what the outcome is. By the way, dogs actually react really well to this type of therapeutic. That's one happy dog. This poor dog on the right was in an accident. The dog has been paralyzed. He actually lost one of his legs and had to go through a series of PRP and stem cell injections as well. And it's very difficult. I was saying this video is too long because it is so difficult to watch any of our fur babies suffer. But you can see here he's starting to walk again. Thinking that there's a leg there, you can see that the dog continues to try to put weight on the leg that is no longer there. I think we've all seen a three-legged dog. We know that they recover from that. But this is what happens in the end. This dog is now home, happy, retrieving balls, and doing really great. You can find IHS Pets at ihspets.com, or you can go through integrativehealthsystems.com in order to schedule an appointment with a doctor to talk to them. This is Dr. Guadarrama. He has over 30 years of experience. He keynote speaks all over the world, and he is just the most amazing person I've ever met. His heart is like the biggest out of all of them. If you want access to the works cited, I'm happy to give it to you. I know you can't possibly, unless you have a perfect memory, look at that and remember it, but you can come to me and we can share the slides. And I just want to say thank you. And this is Strider and Chewbacca. These are my Great Danes. You saw the Great Dane that I have now on the opening slide, and Strider lived to 11 years old, defeating the telomere length, and Chewbacca lived to 10, but I miss them every day. Thank you.
Wow. Yeah, go ahead.
That was great. Thanks, Liz and Bill. So I'm going to take you from the highly sophisticated and amazing and space-age cutting-edge stuff to simple, ordinary kind of stuff that you can implement at low cost. So I'm going to be talking about 4 different things. I'm going to be talking about some simple things that you can do for your own pets at home right now. Some things that are being done commercially to extend the lifespan of dogs by feeding them a certain kind of dog food. And that involves Batman's sidekick, but I will explain in a second. Then there's going to be a little bit of discussion of a possible dog aging intervention trial, which you might be able to help us out with. And then we'll talk a little bit about how we could do maybe something for animals, like Bill Falloon is trying to do for people in his longitudinal aging study. So this is my cat. Her name is Tiggy. It's the only cat I know that sits on her butt. She learned that from us, of course, although we don't do that to the exclusion of everything else. You see Tiggy enjoying sleeping in very peculiar places and just being cute. So we love animals. We love cats. They're unique beings, and we'd like to help them. So it turns out that we give Tiggy 50 milligrams every day of nicotinamide riboside. And the main difficulty of doing that sort of thing is that you can't just go down to your local veterinary clinic and buy this stuff. So we buy ours from Bill Falloon, and then we weigh out 50 milligrams on a little balance. So you can actually buy these balances. They're not very expensive. You can find them online. The one that we have is good to 1/100 of a gram. It goes up to 100 grams. So if you come up with your own idea about some neato thing you'd like to feed to your pet, you can actually do that experiment yourself just by weighing it out and stirring it into their pet food. Now, there's some other things that we do for Tiggy. One of them is we sprinkle carnosine into her cat food. I'm a big proponent of carnosine. I think that carnosine actually prevents your muscles from aging. My muscles are about as strong as they've ever been, and I'm 69, so I attribute that largely to carnosine. So we give Tiggy, we give Tiggy 250 milligrams a day, which is a lot, but I take 3 grams every day, so it kind of works out. You can also give your pet coenzyme Q10. I'm kind of interested in coenzyme Q10 since I kind of introduced that into the United States in a sense. We give Tiggy 5 drops a day of 10 milligram per ml. So that's nice because the liquid form you can just stir into the food. There's also actually a veterinary version of fish oil, and we give Tiggy this one. It contains both EPA and DHA. Now, there's some more conventional sort of prepackaged formulas that you can give your animal. You can just buy them off the shelf. One of them is called Old Friend, and it has some interesting ingredients on the back, but I circled two of them. Ginseng we kind of know about, but I was kind of surprised to find astragalus root in there. I thought Bill would find that interesting. And actually, our cat, as many cats do, has problems with her kidneys, so we give her kidney support. And if you look at the ingredients on that, among various other things, you also find astragalus root in there. So somehow some of Bill's thoughts have been catching on. Okay, so now we're going to go to a commercial product that's out there designed specifically to lengthen the lifespan of dogs. So Burt Ward used to play Robin, the sidekick of Batman in the old TV show. And he's pictured as he is now on the right with two very large dogs, which he likes a lot. And he claims that one of them is 26 years of age and one of them is 20 years of age. And as Bill told you, that shouldn't be possible. Big dogs live maybe 10 years, and that's about it. So if this is really true, it's pretty exciting. The product is called Gentle Giants, and I learned in the course of doing research for this talk that it's actually located in Norco, California. I happen to live like 5 minutes away from this place, so after the meeting's over, I'm going to go investigate these guys. Now, Yuri, our next speaker, pointed out that unfortunately this outfit has gotten some bad reviews. If you look on their look around on the internet, you'll find a lot of one-star ratings for them. So buyer beware. But I would love the chance to sort of investigate it and see if I can determine whether this is bogus or not. Pretty exciting if true, though. By the way, I think I can't tell you exactly why the dog food is supposed to work, but I kind of infer that it's sort of an extreme ketogenic diet. It's about 90% protein from what I can see so far. Okay, so speaking about dogs, I work at during the day. I'm a cryobiologist by day and anti-aging researcher by night. During the day, I work at a company that Bill Falloon provides support for. He also provides support for our sister company, Critical Care Research, which is located in Rancho Cucamonga, California. And Critical Care Research does dog research, and they have a colony of about 31 dogs. Of these dogs, about 12 of them are old enough to be considered getting up there in years. They're about 7 years or more. And so that means that we could start looking at giving them interventions to see if we can slow down or even reverse their aging process. We have good support from Steve Horvath, the developer of epigenetic clocks of all kinds. He has them for humans, he has them for rodents, and recently he developed one for dogs. Fortunately, we may be able to get some support from him it's with some of the costs, but essentially we're going to have to pay $186 per test. We like to do at least 4 tests per dog, and there's 12 dogs, so that computes out to on the order of $9,000 in the end. And unfortunately, we don't actually have any funds to pay for that. We'd also like to include other assays, like maybe NAD assays and things like that. So we need about $10,000 to finish that trial, and as far as what the intervention would be, there's a number of possibilities. There's something called Elixir, which probably none of you have heard of before. It was developed by a guy named Howard Katcher, and he has shown very powerful effects that seem to reverse aging aspects in mice. So he would make Elixir available free of charge for the study if we could pay for the other costs. We have some other ideas about more conventional things, central phenoxine, coenzyme Q10, which, as I mentioned, I'm kind of interested in, and of course, building up NAD levels in the dogs. So if we could get anybody interested in this and help us doing a little bit of crowdfunding to get, say, 10 grand, maybe we could do a dog aging study. Okay, last topic, just an idea at this point. There's nothing really substantial here. It's just that Bill described the virtues of just collecting data on what happens to people as they age, regardless of what kind of treatment they may be on. And you've heard a lot of possible interventions for animals. what if we could set up a similar parallel longitudinal study of aging of animals, just because, you know, their pet owners would like to help us understand how animals age and how we can identify interventions that actually work, which of course, as Liz pointed out, could be a very good model for people as well. So if anyone has an interest in that, we can discuss that later, but that's all I have for right now. Thank you.
Thank you. Hi, everybody. So great to be kind of among the people of your tribe, people who share the worldview of extending lifespan radically, and immortalists and transhumanists. And thank you so much for the wonderful invitation to have an opportunity to tell you a little bit about our work and our work in pets that we plan with a potential gene therapy that I'll tell you just in a minute more details about. But first, here's my pet that I grew up with. He was really kind of like a little brother to me that we unfortunately had to part ways at some point. And fun fact about peptides that Bill brought up, his name is Timka, which is short for Timogen in Russian, which is Thymogen. It's a peptide that my father co-invented with Vladimir Havinson. Some people might know Epitalon, and I'm sure like people here know about this peptide and other peptides. And it's just a funny thing that yesterday I was talking to someone who was just met Vladimir Havinson and used his peptides on his cat, which was 12 years old, and they saw remarkable rejuvenation. Not that I endorse the products of Vladimir Havinson or his approach, but just it's a very interesting fact that, you know, coming full circle talking about my dog, which was named after a product he co-invented. But now onwards to gene therapy. I'm sure many of you have seen this film, The Graduate, and there's this interesting scene where this kind of bit of a crazy uncle pulls away the Dustin Hoffman's character and starts telling him something really important. He says he has just one word for him, and the word is plastics, because this uncle believes that there's a great future in plastics. And some of my friends think that I remind them of this crazy uncle because I have one word that I keep repeating, and in my case, this word is epigenetics, because I think there's a great future in epigenetics when it comes to aging. And here's why. Thank you. Because it turns out that aging can be epigenetically reversed. And first indications of this were shown in 2006 by Shinya Yamanaka, the Nobel Prize-winning scientist, who discovered that these 4 factors, OSKM factors, which were later named Yamanaka factors, can actually epigenetically return a cell back to its essentially embryonic state, pluripotent state, where it is age 0 for a cell. And it's also physiologically rejuvenated while it undergoes this process of reprogramming. And later on, in 2016, a group of Juan Carlos Espiso Belmonte, who got a shout out yesterday, they showed that these Yamanaka factors can actually be triggered in vivo, in the animal. And in doing so, they can also rejuvenate the organism and prolong its lifespan. And this is the study, this groundbreaking study that was published in 2016 by Alejandro Campo and others out of the Belmonte lab. And they showed, if you look at the graph on the right, the survival curve on the right, that the mice that underwent this epigenetic rejuvenation therapy, they lived up to 50% longer. So there are 3 control groups on the left and the therapy group on the right. Now, mind you, these are progeric mice. They have very short lifespans, but still, this kind of first proof of concept that this can be done safely, I think it's remarkable. And this is what inspired me to start Eutherium back in 2017 to translate this therapy into something practical that we can all use for ourselves for radical life extension. Why is this even possible? Why is epigenetic rejuvenation possible? Because it turns out that we're lucky that the cellular reprogramming happens gradually. It's not like a skin cell just jumps back into becoming a pluripotent embryonic cell. It does so gradually while being rejuvenated in the process. And this kind of inspires us that we can find the therapeutic window in which it is still, the cell doesn't lose its identity, it is still a skin cell, but it is remarkably rejuvenated while remaining the skin cell that it started with. So there's this slide that shows this gradual process, and it has become very popular. Steve Horvath has shown this slide a number of times. Michael West also uses it in his presentation. This is just a graphical representation of what is happening while the reprogramming is going on. So as OSKM genes are being induced, the epigenetic age, the blue line, gradually decreases, while cell identity also decreases gradually. So potentially this narrow blue window, if we can hit it repeatedly, we can enjoy rejuvenation without losing functional status of the cell. And this is kind of the holy grail that the epigenetic rejuvenation companies are now trying to explore. And thankfully, it's not just us these days. There's several companies that have entered this field since 2017. I think the first one was Turn Bio out of Stanford, out of the lab of Vittorio Sebastiano and his student Jay Sarkar, who in 2015, I think they published the paper Rejuvenation on the Road to Pluripotency, which showed this proof of concept that this is possible. And then, of course, the Belmonte paper came out. Now, all of these companies exploring epigenetic rejuvenation, they all have subtle differences. For example, Turn Bio, they're focusing on, first of all, they use 6 factors, not just Yamanaka, but also 2 other factors. And they're looking at ex vivo rejuvenation. And also they don't want to use not genes, but mRNAs, which is, they think is a safer approach. Then, of course, there's David Sinclair, who's now, I'm sure you've all heard, is now also exploring this epigenetic reprogramming approach, kind of off from his NID. And they're using 3 factors, OSK factors without the MYC. But now they're using actual gene therapy. They're using AAVs to deliver locally these Yamanaka genes in the places of interest. For example, into the eye, they want to rejuvenate old eyes and maybe fight glaucoma, and also trying to rejuvenate nerves in case of paralysis. And they're saying they will enter clinical trials maybe as early as next year. For this, they formed this company called Iduna or Iduna. Iduna is the Norse goddess of rejuvenation, just a piece of trivia. And he kind of attracted all the heavy hitters from the field, starting with Juan Carlos Espiso Belmonte, Steve Horvath, and Manuel Sorano, who was also one of the pioneers of in vivo reprogramming. And so they're exploring that paradigm. Also, there's Michael West, who I'm sure you all know very well. Now, out of Ajax, they've spun out this reverse bioengineering company that will focus on this epigenetic rejuvenation approach, probably using the small molecule cocktail, but I'm sure Mike will tell you much more about it tomorrow while he will be speaking. And finally, this is us. We will pursue actual in vivo gene therapy. We would like to get the necessary genes into the organism, and then these genes will be normally silent until we activate them with some kind of inductor. Right now, they use doxycycline, which is used in many models to activate these genes, but we really want to move away from an antibiotic, something more inert or maybe even a novel patentable molecule. And PETs are a very interesting strategy for us because they provide an opportunity to circumvent the delivery problem. Because of course you have to get all the genes into the cells of an organism, and for an adult you need to target trillions of cells, which is an issue in itself. But if you can do a GMO PET, a transgenic PET, into which you insert those genes while still an embryo, those genes get copied into all the cells of an organism, and you can activate them and experiment on the PET, seeing what kind of life extension it can obtain from using these genes. So when can you expect these therapies? For GMO animals, within 5 to 6 years, if everything goes according to plan and we don't have any kind of setbacks, maybe safety issues. And then for existing animals, genetic therapies, we approximate that within 8 to 10 years, you'll be seeing it on the market. And for human therapies, it will probably take another 8 to 10 years. So just one parting word from me. You guessed it, epigenetics. There's a great future in epigenetics, so just think about it. Thank you.
All right, is my headset working? Yeah, okay, so we have a bunch of questions here. This first one's not really a question, but I think it's a really good thing, so I'm going to go first with it. It's from somebody that says, I own Plants for Pets, a company that uses some of its profits to help pets in various ways. We'll cover the $9,000 to get the mentioned dog trial started.
Thank you.
Please
Thank
let us know
you.
how to do so.
Wow.
So
Woohoo.
whoever that is,
All right.
see Greg.
Wow.
Thank you. Thank you. Thank you.
Thank you, thank you, thank you. And I'm going to get it on Dash too. So another question to Greg. Can you talk more on NAD for animals?
Well, a little bit. I don't know of any studies that have actually been done trying to supplement NAD in pets yet, but we do know something about supplementing NAD in lab animals. And we did hear a wonderful talk yesterday about an even more powerful way of supplementing NAD that may be applicable to pets as well. So I would encourage everyone to look into that. But David Sinclair came out with this revelation in 2013 that and he went into incredible detail about all the biochemistry, and then it all built up at the end of the paper to this experiment they did in which they just fed nicotinamide mononucleotide to the rodents that they were looking at, and a week later, every measure that they had of aging had reversed back down to the youthful state, just with NMN. NMN is something that you can buy. I actually plan to switch to using it myself. I'm taking nicotinamide riboside now, but I'm going to switch over to NMN because all of these studies in animals that have reversed aging by supplementing NAD levels with precursors have used NMN, not NR. There's nice studies in humans with NR that you can raise your NAD levels in your blood with NR, but the studies that really showed rejuvenation in animals have been with NMN. Now, the problem with that is that to get the human equivalent of NMN, you would have to take about 60 grams of it a day. So there is a limitation on that, and I would encourage anyone who wants to try this on their pet to consider any ways they can to further augment the results. But NMN might be the best way, but NR is perfectly valid as well. And once we get to the point where we can actually measure, say in a blood test, how your NAD levels in your pet are responding, then you'll be able to sort of decide how effective your intervention is.
Now we have about 5 more questions, so I'm going to ask that we spend about 1 minute on each of them because we're going to run out of time. Are any of this age reversal, or just age prevention? Let me start off with that one. What I'm trying to do is I'm trying to get age reversal, but we don't have it done yet until we actually start seeing if it works. But the approach that I am working on has been shown to reverse aging in mice, engineered mice that have been shown to age, that have been engineered to age by telomere shortening. They actually, Dr. Rhonda Pinnell actually saw what he called a remarkable reversal of aging process in a lot of ways, not just energy, but a lot of different ways. Maria Blasco has done a lot of work. Most of her work is actually showing prevention of aging. Treated animals didn't age as fast as the untreated animals. But it's still a sign that reversal of aging by telomere shortening could be possible. Anybody else want to comment?
I would agree with that. So all of the genes that we're looking at, the 3 genes that I told you about, the Klotho, the follistatin, and the telomerase induction, they're all genes associated with extending lifespan in animal models in research. So does that mean that we are slowing aging? Yeah, probably it does. And what will we need to do to stop and reverse it? Well, those genes are associated with that. They do affect the hallmarks of aging in some ways, but we need to find a multi-combinatorial effect in order to actually reverse it significantly. But again, the gene therapy you're working with and the gene therapies, all of the gene therapies we're working with are all associated with extending lifespans. As a matter of fact, follistatin extended lifespan in various animal models, and it's actually already passed through safety and efficacy for humans for muscular dystrophy, and so we will see in our patients that are aging if we can extend lifespan with it. But as far as full body age reversal, that's something that we're working hard on in research and development in our company, and that's what we're all looking to do.
All right, and anybody else on that?
I would just support what you said, Bill, and of course what you said, Liz, but in that study that you referred to, one of the effects that they saw was a regrowth of the brain.
Yeah.
You know, I mean, that's pretty impressive. So that's reversal. That's reversal.
Yeah.
Yeah.
Anything?
Well, it just be a reversal. Maybe it's a short-term reversal. It's not the reversal that we kind of all think of when we think about living indefinitely, but it's just a short-term reversal in experiments that hopefully at some point we'll be able to do in a more sustained manner.
Yeah.
Next question. Is there a difference in therapy for different animals? Responsiveness, application, et cetera. Let me start again. We have found at Sierra Sciences that human telomerase does not work very well in dog cells. But dog telomerase works really well in dog cells. Dog telomerase doesn't work well in human cells. And we can say the same thing about cats, horses, and pigs too. They don't cross-react very well. So we have created gene therapies specifically for each animal. We've isolated the telomerase gene for each animal, created the gene therapy for that. Anybody else want to comment on that?
No, we do the same thing. We specifically engineer the animal's version, that species version of the gene that would be beneficial for the animal itself.
I guess the only thing I would add is that, at least with respect to the NAD story, that's probably a universal aging process. So it probably would affect all animals and humans in a similar fashion.
Right, it's a general thing for gene therapy. It has to be species-specific genes that you're delivering. So, yeah.
Okay, next question. They said C60 olive oil doubles the lifespans of rats. Can we give C60 olive oil to our dogs? Well, first, I know a lot of people that do. But anybody want to comment on C60?
I have not really looked into it. I know the legends about it, but I haven't actually read the literature.
Yeah, I knew all about C60 long before it was ever considered a drug. It was a big goal for chemists to try to design what's called a buckyball and then actually turn it into a bucky tube. But when it actually turned out to be a drug that might benefit animals and humans, I was really surprised by that. Does anybody else want to comment? What is the rapamycin dose for pets? What is the formula? Wait, I talked about rapamycin. Anybody else talk about rapamycin? I don't know. It's published in a paper, and hopefully if you get copies of the slides, you can go look it up. But I know George Church and his scientists are working on that.
Yeah, and the University of Washington is also doing a study, and they did have it published because I saw it online.
Good. And that's actually one of the authors, Promislo, I think, was at the University of Washington there. Is there any beneficial treatment for dogs with seizures? Okay, anybody? I have no idea.
Yeah, I wonder if that would be why the dog was having a seizure, but
I mean, presumably there are conventional treatments like Dilantin or whatever your best choice is for an anti-seizure medication. So conventional medicine might be the answer there. It'd be nice to prevent the cause of the seizure, but I'm not quite sure how to do that.
it's a great question.
Last question, actually a new question got asked. Can exosomes be used for dogs for rejuvenation? And we are using exosomes, so is Mike West. Mike West is using exosomes, and we're using exosomes to deliver telomerase to cells too as an alternative to the other types of gene therapy. But exosomes actually provide apparently a lot of benefits even without delivering other genes. So I think exosomes is something that's going to get really popular in the near future, and I don't think enough studies have been done to really answer the question yet. Anybody else want to comment?
Right, again, maybe need to be species-specific exosomes,
I would
but
bet you money
could
it's
be even
species,
some
yeah.
cross-species benefits.
And I guess that's it. Thank you very much. I hope you enjoyed this and we can have it back next year.